Hello everyone. This is your bio coach, Dr. Shilpa. Today's topic is biological classification. So, which all scientists come to your imagination when you think about biological classification? We would be reading about three: Aristotle, Carolus Linnaeus, and R.H. Whitaker. These classifications help us to actually categorize all the living organisms into various ranks for easy identification and easy studying of them. Now, the first classification system that was given by Sir Aristotle was based on morphological features. Why do you think that was? Because at that time, microscopes were not used to be present. And that's why he could base his theory on his observation. Observation means what he saw physically. He saw the morphological characters of organisms. And that's why he divided all the beings into two: plants, who didn't used to walk and didn't used to eat, and the other one was animals, who used to walk and used to eat from outside. Plants were further divided into trees, shrubs, and herbs, while the animals were divided into red-blooded and the cold and the non-red-blooded ones. Okay, we would not be reading about classification system given by Carolus Linnaeus today. This other classification, which is the most popular one, which we read even in the current times, is a five kingdom classification, and it was given by Sir R.H. Whitaker. Now, the previous one was based on the morphological characters, whereas this one was based on a comparative account, because by then we also understood the concept of evolution. Evolutionary relationships are phylogenetic relationships. Now, to help you remember, this word comparative can be divided into three syllables: comparative. From com, you just take the first and the last alphabet, C and M. From para, you take P and R, first and third. And same in the last. In tiv, you just take first and the last, T and E. So by C, we mean cell structure. By M, we mean mode of nutrition. By P, we mean phylogenetic relationships. By R, we mean reproduction. And by T, we mean thallus organization. This last E is the same as phylogenetic relationships. By E, we mean evolution. Okay, this should help you to remember and retain this part, that the five kingdom classification was given on the basis of comparative features. And the comparative features were these five. Let us start with kingdom Monera first. You know that in kingdom Monera, unicellular beings are there. Now this image should help you remember that there is an arrow and it is on an aim. So AEM, not AIM. And it is there in this name also. You see the last alphabet of Monera, A. In between you see E, and then the first one here you see M. So archaebacteria, eubacteria, and mycoplasma. The first category, archaea, it means ancient. These bacteria, since they are called as ancient, they were present in the ancient times and they are present from the ancient times. So they are habitual to living in extremes of environment, extremes of temperature, pH, light, salinity. Okay, all such kind of bacteria would be the RK bacteria. Then EU means true or good. So they are the bacteria having a true cell wall, a rigid cell wall. And then myco, like microbes that we are studying about. So these mycoplasm are the smallest known living organisms to humankind so far. Okay, so this mnemonic AEM should help you remember the categories of kingdom Monera. Today we would only be discussing about eubacteria. Eubacteria are the true bacteria having a rigid cell wall, which you can see in this photograph, the halo effect around the bacillus. Just by this, you can remember that they have a rigid cell wall. Rigidity means stiffness. And by stiffness, you should be able to remember cane. This cane is actually the mnemonic for the 4 examples of eubacteria. C. Cyanobacteria, which are the blue-green algae. A and N, nostoc and anabena, which are the nitrogen-fixing microbes. They fix nitrogen from the soil. They fix nitrogen from the environment into the soil. And the last one, E. coli, Escherichia coli, which causes diarrhea in humans. Now, if you put this cane in a mirror image and incline like this, it forms A. It should help you remember the other category or the other basis by which you can divide these bacteria. The first one we remembered, the 4 examples by the cane. The second A should help you remember autotrophs. So we are actually classifying eubacteria into phototrophic autotrophs, heterotrophic autotrophs, and heterotrophs. So the phototrophic autotrophs are that example of CAN, cyanobacteria, nostoc, and abena. The chemosynthetic autotrophs are methanogens. They actually help in nutrient recycling, such as of sulfur, iron, phosphorus. Then the heterotrophs. Heterotrophic eubacteria are the 2 types. You can remember the ones which are beneficial to living organisms and to humans, and the ones which are not beneficial, the ones which are harmful. We call them as pathogenic. So these 3 photographs, they show the examples of pathogenic bacteria. In the first picture, you see the back of a person having red spots. These are called as rose spots. It is the classical feature of typhoid fever caused by Salmonella typhi. The second is a picture of a choleric patient. The patient looks weak, fatigued, must have lost a lot of nutrients from the body, and that's why he looks like this. So cholera is caused by Vibrio cholerae. The third is a classical sign of stomatitis of tetanus. Tetanus is caused by tetani, Clostridium tetani. Okay, and then there are good examples of bacteria. Some help in conversion of curd, in the formation of curd by the conversion of milk. Those are lactobacillus. Then there are those which help in antibiotic production, such as penicillium to make penicillin, streptomyces greases to make streptomycin. Then there are the ones which are present in the roots of leguminous plants, and then they fix nutrition from the soil into the plants, into the roots. And the example is rhizobium. Okay. We discuss that eubacteria are photosynthetic autotrophs, cyanobacteria, nostoc, and abena, chemosynthetic like methanogens, and then heterotrophs. And in heterotrophs, we read about the pathogenic ones, uh, such as typhoid, cholera, tetanus, In this, I would also like you to remember the example of mycobacteriums. The tubercular one you know about, it causes tuberculosis in humans, especially in hot, moist, humid environment like like in slum areas when people when people reside all together, then they are the most common. This is the most common disease that can happen there. And then there is another kind of mycobacterium, which is Mycobacterium marini or marinum. They actually affect the body of humans having weakened immune systems, such as in HIV patients. Okay. Then these are pretty common examples. Psittacosis you must have heard about. It is caused by parrots or other birds. So these birds are actually carrying the microbe, this eubacteria, a heterotrophic type of eubacteria, Chlamydomonas psittaci. Then there in cats, there is Bartonella henselae. Okay. And in plants, there is an example of citrus canker, which is caused by Xanthomonas citri. Okay. I think that these mnemonics would help you to remember the material, the content, and even these pictures. Now, getting on to the kingdom protesta, since earlier we were talking about walking, now we are talking about being pro, being skilled at walking, like being an expert. So earlier we were talking about walking on a flat ground with a cane. Now the mnemonic is walking vertically on the steps. So instead of ST, we are using CD. CDEPS, okay? Chrysophytes, dinoflagellates, euglenoids, protozoans, and slime molds. The chrysophytes, this is the one category that we would be discussing today. Chryso means golden. So that's why these are golden bacteria. They are, sorry, they are not bacteria. These are called as golden algae. They are diatoms and desmids. Both are microscopic and they move because of water currents. So they float in water passively. Okay. Diatoms, especially, are the chief producers in the ocean. They are the autotrophs and they are present in most abundant quantity. Now, with this name, you can remember that diatoms having two atoms. So having two shells which which overlap and then fit together in a pattern which looks like a soap box, as you can see in the first example. And that's how they have gotten this name of diatom. Moreover, they have silica embedded in their shells, and that makes them indestructible. So they keep on accumulating one over the top of each other. And that's how we get the diatomaceous earth. This earth is coarse in nature, it is gritty in nature, and hence it can be used for two purposes, polishing and filtration. Polishing you can easily understand, but how does filtration occur? It occurs by the phenomena of adsorption. So now here in this photograph, first photograph, you can see a diatom molecule, a solid molecule. Now this adsorption is the phenomena that one single solid molecule would be able to attract small liquid or gaseous molecules. So here we are talking about oil, which is a liquid molecule. And then they form a thin layer all around them. And that's how oil gets filtered and the impurities are taken away. And this is the another application. The third application you can remember by this photograph, that around the plant, this diatomaceous earth is being spread around in a circular fashion so that it would keep the pests away. Okay. Desmids, golden algae, and they are the same as diatoms, that they are microscopic and they float passively in water. Now the third category, kingdom fungi, that you can remember by this photograph. The mnemonic is that you see this red colored mushroom. It appears to be dangerous, so it is bad for people. And you know already that fungi, some are good for people and some are bad because they can be poisonous for people. or for other beings, living beings. So the mnemonic is BAD and P, bad for people. And here we would be studying only about the Ascomycetes today. Okay, so before we get onto the Ascomycetes, I would like to share a few terms which are common amongst fungi. When we talk about fungi, we talk about these terms, but then the students, they're not clear about these terms. So in fungi, just remember, if a thread-like long, slender thing is there, that is just an hyphae. And when this hyphae is present in a network form, then it is a mycelium. Okay. Now, the third term is coenocytic hyphae. When you see these thread-like structures having nucleuses, the dots inside them, but there is no separation between the dots. There is no wall between these dots, between these nucleuses. Then that is a coenocytic or non-septate hyphae. And when these boundary walls are present, then they are called the septa. The walls are the septa. So that's why such hyphae becomes septate hyphae. Okay. Now the Ascomycetes. They are basically the sac fungi. In the name also, if you rearrange the first three alphabets, you get the name sac. So you should be able to remember they are the sac or the perfect fungi. Even Basidiomycetes are the perfect fungi. Deuteromycetes are not. Okay. Now here in the first example where we see orange mushroom, we see that it is growing on a decomposing body. And that's why we call this fungi as saprophytic. Whereas in the other figure where we see white mushrooms, it is actually growing on dung. The next figure also shows dung and it has these microscopic fungi over it. Both of these are the example of coprophilous fungi. Coprophilous means dung loving. And the example is Pylobilus. In the second image, the example you see is of Pylobilus. Pylobilus is one kind of coprophilous fungi. Pylobilus is also an English term, and is this name is given to dance formation also. When multiple humans are in a formation where they are lying on top of each other, they are standing on top of each other in a diverging manner in a whole circle, then that shape is this. Because as you see these small round balls of fungi over this dung particle, when they blast, when they burst, the material inside, the material which is present inside, it just comes off and gets spread everywhere. So this divergence leads to this name of Pylobilus. Example of saprophytic fungi, Candida albicans, is also pretty common, and you would be studying about it during your medical courses. So you know that albinism is when we see a person and he or she appears to be white in a specific way. Uh, they are not lighter or darker skinned, but we call them as having white patches. So that whitishness is because they lack melanin pigment. So that white means albinism, and this albicans, this term comes from there only. So you should remember that Candida albicans is an opportunistic fungal infection which happens on tongue, and it looks whitish in appearance. The last two photographs show you that. Well, this ends here. See you in the next one.